So we've been very interested in the chemical control of microbes since the 1800s. Back then, we used chloride of lime and iodine to clean wounds and wash hands before surgery. Now, we seem to have a preoccupation with eliminating microbes. It actually borders on excessive. We deodorize, we sanitize, we kill germs, we disinfect, blah, blah, blah. We seem to have this obsessive fear of germs and need to kill them. Industry really capitalizes on these fears. Look at all those commercials out there for antimicrobial, this, that, and the other. But there's a problem here because this could result in favoring the growth of resistant bacteria. Not only that, it's already evident that Staphylococcus bacteria show increased resistance because of all these antimicrobial products. Studies are even showing now that when bacteria become resistant to these antibacterial agents, they also become resistant to antibiotics. So it's just not the overprescription of antibiotics that's causing this great resistance. So even though I know from taking this class was seeing that there's bacteria everywhere, I urge you to really be conscious about how and when and why you might be using antimicrobial products. There are approximately 10,000 different antimicrobial chemical agents being manufactured these days. Approximately 1,000 of those are routinely used in healthcare and at home. They can occur in liquid, gaseous, or solid state. Now, most often, those that are found in solid and gaseous states are dissolved in either pure water or in water and alcohol mixes. Aqueous solutions are just pure water, and then tinctures are solutions that are dissolved in pure alcohol or water and alcohol mixes. Here are some of the chemical agents that we use in healthcare. You don't need to spend too long looking at this table in detail because as we go through the rest of this lecture, we'll cover most of these, but this is a great summary table for you to study from. So again, choosing to use and understanding the appropriate antimicrobial chemical to use should be of constant concern, especially in medicine and dentistry. Here we have a list of some of the desirable qualities. As yet, no chemical actually fulfills all of these, although glutaraldehyde and hydrogen peroxide come the closest. When choosing a microbial chemical, we'd like it to have rapid action, even at low concentrations. Also, solubility in water or alcohol, and long-term stability in that solution. We'd like broad-spectrum microbicidal action without being toxic to human and animal tissues. That's a tough one. Also, we like the chemical to penetrate the inanimate surfaces and sustain cumulative or persistent action. Here's another big one. They need to have resistance to becoming inactivated by organic matter. The issue with organic matter is that large amounts of it, like feces, saliva, urine, or pus, can actually hinder the penetration of disinfectants into the surface they're designed to cleanse. Sometimes these compounds can even form bonds and reduce the activity of the chemical agent. Ideally, we have non-corrosive and non-staining properties, sanitizing and deodorizing properties, and they're affordable and readily available. Now, naturally, there are some factors that affect the germicidal activity of chemicals. First of all, the nature of the microorganisms being treated. Are they vegetative, less resistant microbes, or are they endospores? The nature of the material being treated, whether it's porous or solid surface, the degree of contamination, the time of exposure, and then the strength and chemical action of the germicide. How does it work? And how much is required? So you can see over here on this table on the right side, there are several different agents that are outlined here, and it tells us how long each of them take and what concentration is necessary for the destruction of these microbes. I'd like to draw your attention here to the hydrogen peroxide. You'll notice that it has very fast action. 3% solution is only necessary, and within a matter of seconds, we can take care of the majority of microbial contaminants. So now I'd like to take a look into some of the germicidal categories and chemical groups. First, we'll take a look into the halogen antimicrobial chemicals. These are things like fluorine, bromine, chlorine, and iodine. They're non-metallic elements, and they're all in the 7th group of the periodic table. So you see them here. So they have similar chemical behaviors. They are both microbicidal and sporicidal with longer exposures. All of them are highly effective components of disinfectants and antiseptics. Chlorine kills both bacteria and endospores, as well as fungi and viruses. A great example is our use of household bleach. It's less effective if it's exposed to light or alkaline pHs, so things that are alkaline are like dish soaps, other detergents. This is why it's really important to make sure that we rinse dishes between using soapy solution water and then our bleach water for sanitation. Another thing that prevents chlorine's action is excess organic matter. Next, we'll look at iodine. Iodine can be used as topical antiseptic or disinfectant. It's not adversely affected by organic matter and pH. Strong iodine solutions, like 5 to 7% or tinctures, are really no longer considered safe as routine antiseptics because they have a high irritation factor for the skin, and they're actually toxic when they're absorbed. So if we use them in high quantities on the skin, then that substance can be absorbed and form toxicity. Iodine kills all classes of microorganisms when given the proper concentration and exposure time. More commonly these days in medicine, we use iodophores, which are complexes of iodine and alcohol. These are found in solutions like betadine and isodyne and are used in medical antisepsis because they're much less irritating to the skin. Another thing we can use alcohol for is disinfecting water during emergencies. So both chlorine and iodine can be used to disinfect water. Chlorine, in fact, is used to disinfect most of our drinking water. However, it's used at a very, very, very low concentration, about 1 part per million or less. And probably many of you have used iodine also in the backcountry instead of carrying a water filter. You can even buy little tablets to drop in 1 per liter. Otherwise, just 3 drops will work. The next class of germicidal chemicals we'll look at are phenol and its derivatives. Now, phenol itself is a very poisonous compound, and it was derived long ago from the distillation of coal tar. It was the major antimicrobial chemical before other less toxic phenolics were developed. There were hundreds of them developed. So now we actually compare each of those phenolics against phenol itself. The phenol coefficient is the measure of this, where we're comparing the chemical's antimicrobic properties to those of phenol. In higher concentrations, these phenolics are actually cellular poisons, but at lower concentrations, they can inactivate certain critical enzyme systems and be more microbicidal. Keep in mind, though, that phenolics are not reliably sporicidal. Here's your basic phenol aromatic ring structure. As you can see, there are a number of different arrangements of these phenol structures. Hexachlorophenol and chlorophenol are phenolics. In fact, hexachlorophenol is a bisphenol. Bisphenol is one of the components that's found in Lysol. Another very commonly used phenolic is triclosan. It's the most widely used. It's found in soaps, kitty litter, sponges, laundry aids, so many of the products that we consume that are advertised as antimicrobial. Phenolics act well in the presence of organic matter, and they have great detergent actions. However, their toxicity makes them much too dangerous to use as antiseptics, so we would not put them on our skin. Chlorhexidine is another class of antimicrobial chemicals. It's sold under the brands of HibiCleanse, Hibitane, or Peridex. It's a complex organic base containing chlorine and 2 phenolic rings, so that same phenol structure, and it targets cell membranes and protein structure. So as the cell membrane weakens and the enzymes are denatured, then the microbes are destroyed. At moderate to high concentrations, it's bactericidal for both gram-positive and gram-negative bacteria, but it is inactive against spores. It has mild to low toxicity as well as very rapid action. It's used in hand scrubbing, preparing skin for surgery, obstetric antiseptics. It's even used as a preservative in eye solutions or contact lens solutions. It's used as a wound irrigant, and it's also put into many mouthwashes. You'll notice as we move through these various chemical microbial control agents that many of them are found in a lot of our household products. And honestly, it makes me a little wary, because if they do have antimicrobial activity, then they probably have some effect on our cells also. But I guess, as they say, you have to pick your poison. You're probably aware that alcohols can be used as antimicrobial agents. Isopropyl alcohol and ethyl alcohol are really the only ones that are suitable for microbial control. And their mechanism of action really depends in part upon their concentration. Alcohol at a greater than 50% solution will dissolve membrane lipids. Once we get up to 50 to 95%, it denatures proteins. Now, alcohols are one of those things where it's not the more the merrier, as in the more concentration there is, the more destruction, because once you get above 95%, you actually don't see denaturing of proteins, but rather just dehydration. That's because alcohols depend upon an aqueous solution of some sort in order to denature the proteins. Now, alcohols do not destroy bacterial spores at room temperature, but can destroy very resistant vegetative bacterial forms. They are most effective in inactivating enveloped viruses rather than non-enveloped viruses. And then isopropyl alcohol is more microbicidal, but also much more toxic. In fact, it has adverse effects on the nervous system if inhaled. This is something to keep in mind because isopropyl alcohol is the alcohol that's sold on the grocery store shelf as rubbing alcohol. So don't inhale that stuff. Now let's take a look at hydrogen peroxide. It's exceptionally effective. Hydrogen peroxide has its germicidal effects due to the direct and indirect actions of oxygen. And it actually decomposes in the presence of light to water and oxygen gas, so fairly innate substances. However, the oxygens tend to form these hydroxyl free radicals, which are highly toxic and reactive to cells. Much like free oxide radicals, they attack various components of proteins, so they can be very harmful to tissue. And a 3% solution is plenty for most needs, like skin and wound cleaning, for mouthwash. It's even effective for soft contact lens sterilization. Hydrogen peroxide is very effective for the rapid sterilization of expensive tools. This machine here is designed for rapid sterilization of endoscopes and other microsurgical instruments that are not cleaned effectively or efficiently by any other means. Hydrogen peroxide is bactericidal, virucidal, fungicidal, and at high concentrations, it's also sporicidal. So hydrogen peroxide is just an amazing germicide with very few bad side effects. We can even sterilize whole rooms with hydrogen peroxide vapors. Next, we'll look at chemicals with surface action, like detergents. They act as surfactants, and we've already explored surfactants. They are amphipathic, right? They have two different ends. Just like a phospholipid in a membrane, they have a charged end and then an uncharged end. So they have a polar region and a nonpolar region. Now, they can either be cationic or anionic. The anionic varieties are less effective, but cationic detergents are very effective. The positively charged end interacts with the predominantly negatively charged bacterial surface proteins, and then the hydrophobic or non-polar hydrocarbon chain tail is able to interact with the membrane phospholipids itself and disrupt membrane structure. So these amphipathic molecules are able to very easily disrupt cell membrane. However, they have a greatly reduced activity in the presence of organic materials, so excess saliva, pus, serum, so on and so forth. Now, soaps are weak microbicides, but they gain much more germicidal value when they're mixed with other agents, like chlorhexidine or iodine. Next, let's take a look at some heavy metal compounds. Heavy metals have high atomic mass, and they include mercury, silver, gold, copper, arsenic, and zinc. However, only preparations containing mercury and silver have any significance these days as germicides. Each of these heavy metals, though, have oligodynamic action. That means they have antimicrobial effects in exceedingly small amounts. You can see over here in this petri dish that just these small specks of either silver or gold have caused there to be no microbial growth anywhere near the region of that speck of metal. The way that these metals work is by binding onto functional groups of proteins because they're charged and thus inactivating them. This destroys most microbes, but not endospores. Now, naturally, there are some drawbacks to using metals in microbial control. First of all, they can be extremely toxic to humans. Think about what you know about mercury. They can cause allergic reactions. Large quantities of biological fluids and wastes actually neutralize their actions. So again, organic material neutralizes the actions of heavy metal compounds. And then microbes can also develop a resistance to them. It's interesting to note that silver ions are now being incorporated into hard surfaces like toilet seats or stethoscopes, cutting boards, refrigerator doors, and they're incorporated because they are able to control microbial growth, just as they have in the petri dish on the right. Before we move on, let's have a quick concept check. What kind of property do surfactants have? Now, we've covered this a couple of times. Surfactants or detergents, are they hydrophilic? Are they hydrophobic? Are they amphipathic or hypotonic? Well, hydrophilic, we know, water-loving. We'll cross that off the list. Hydrophobic, water-fearing. We'll cross that off. Hypotonic, that's not a relevant term. Amphipathic is the correct term here, because amphipathic has both hydrophobic and hydrophilic groups by definition. Now a look at aldehydes as germicides. All aldehydes have a CHO functional group on their terminal carbon. This is a really strong reducing group. That is, it takes electrons. Sugars and some fats are aldehydes, but that's not what we're concerned with here. The two aldehydes that we really will think about in microbial control are glutaraldehyde, which we've heard a little bit about already, and formaldehyde, which, as you know, is used in preservation of tissues and thus has some issues. Either way, the aldehydes work by forming cross-linkages between proteins on the cell surface. So here you can see that it's linked up this protein with this protein. Or in a gram-positive situation, it'll grab onto amino groups in the peptidoglycan cell wall. Naturally, this causes some issues with the integrity of the cell membrane, as well as how some of the enzymes function once they're inside the cell. Glutaraldehyde is the most commonly used, and it's often used in conjunction with autoclaving, as we've seen previously. Now, formaldehyde is a really strong carcinogen, so we definitely don't use that as much as we used to. There are several gaseous sterilants and disinfectants. Three of the most commonly used ones are ethylene oxide, propylene oxide, and chlorine dioxide. Both ethylene oxide and chlorine dioxide are great sporicides. Ethylene oxide is mostly used in sterilization of prepackaged medical devices, plastic materials, or disposable petri dishes, those sorts of things. However, it has issues because it's highly explosive. It needs to be used in conjunction with other gases to make it safe. Also, if we are exposed to it, it's highly carcinogenic. Propylene oxide is much less toxic. It's used for sterilization of foods, like nuts, powders, and spices. Chlorine dioxide is used for the treatment of drinking water worldwide, as well as wastewater and food processing equipment. The way that these sterilants work is by blocking DNA replication and blocking protein function. Gases can provide a versatile alternative to heat and liquid chemicals, and are especially useful for substances that cannot tolerate the moist or dry heat antimicrobial mechanisms. Next, we take a look at dyes as antimicrobial agents. We're talking about dyes like crystal violet and malachite green. Many dyes are the primary source of certain drugs that are used in chemotherapy. There are aniline dyes, which are very active against gram-positive species of bacteria and various fungi. For example, aniline dyes are like a combination of crystal violet and malachite green are used in the treatment of ringworm. Then there's yellow acridine dyes, like acroflavine and proflavine, sometimes used for antisepsis and wound treatment. There are limited applications for dyes because they stain and they have a narrow spectrum of activity. Who wants everything to be green or violet colored? You're probably also aware that acids and alkalis can be used as antimicrobials. Very low pH or very high pH can destroy or inhibit microbial cells that live in our realm. These types of treatments are limited due to their corrosive, caustic, and hazardous nature. But here are some common ones. Aqueous ammonium hydroxide is a component of many of our detergents, deodorizers, and cleansers. Organic acids are used in many food preparations. Acetic acid is a pickling agent. Propionic acid, lactic acid, benzoic acid, and sorbic acids are used as preservatives to prevent contaminations of food by fungi and vegetative microbes. This is a great table that summarizes some of the main ingredients in many of our antimicrobial products. Take a quick peek and see if you recognize any of those that you use yourself. That brings us to the end of this chapter, but before we move on, let's have a quick concept check. Which of these would be the least likely to be removed by passage through a HEPA filter? A virus, a yeast, a bacteria, or a protist? Well, the question simply here is which is smallest, right? So definitely we know that viruses are the smallest of these.